Great. Okay. Well, thank you very much for the invitation. Thanks, everybody, for showing up this morning. So I'm going to take a little bit of a step back, and instead of focusing on anatomy, I'm going to be very much focusing on some of the work we've been doing for coming up with methods for crossing scales. I'm almost a little surprised we didn't actually see something like this yet today, but oftentimes when we talk about challenges of scale, we show something like this, demonstrating that the structure and function relate to each other over many, many orders of magnitude in the brain. We all know this. The reason why I wanted to show this is because I actually increasingly do not like showing this kind of a depiction, because it sort of places MR way at one end of what is being laid out for you is the goal. And although, of course, we are sensitive to various aspects of microstructure using MR, increasingly, we at our center like to sort of think of it more in these terms, which is that neuroimaging is kind of a almost completely unique tool. There are very few things that allow us to really integrate across many, many levels of investigation using the same tool in neuroscience, from everything from small tissue samples, which I'll talk about today, to rodents where we can do causal manipulations, to large populations, and even all the way up into individual patients. And so increasingly, I like to think of it in terms of ways that we can sort of bridge across these scales using a single tool. So the approach that we've been taking mostly in my group is to, which we've heard a lot about today, is to try and relate in vivo MRI all the way through to optical imaging. And as we've heard a lot today, postmortem MRI is particularly important, and I think it's worth stating why postmortem MRI is really powerful. It shares common signals with what we get with in vivo MRI, potentially in many, many thousands of individuals, but it shares common tissue state and also common tissue with other techniques such as optical imaging. So I'd like to very briefly talk about some of the technical challenges. These have been kind of touched on already, but just to be very clear, there are real technical challenges to doing these kind of experiments. First of all, you have to handle actual specimens, so you have to do things like susceptibility match. You need to come up with nice ways of holding brains to get consistent placement and good shim quality, and all of this is a lot of work to actually get your sample in a form that you can actually get good data in it. And one of the things that we've encountered is that people oftentimes sort of give up quite early because they actually haven't gone all the way toward achieving these sort of careful sample handlings. Another challenge that you face is that postmortem tissue is different. So this is a beautiful example from Tim Darby, where he was able to scan a macaque brain using a rodent scanner, and the simple issue that the macaque brain fits into this scanner makes things much easier. If you try to do something similar on a whole human brain in a human scanner using the in vivo techniques that have been work really, really well for us there, you'll get data that looks something like this. So within my group, what we're using is a technique that Anastasia actually touched on earlier called SSFP. You can get really beautiful data down to, for example, here, 0.5 millimeter resolution. Now that said, here's another challenge. It takes a long time to get these data sets. So this was a 5-day scan. At this point, I think we could get this data probably in a weekend. You can start to see really beautiful images coming out of it, but it's also clear that it's going to be a relatively limited number of data sets that we're going to be able to do this on. So that's another challenge. Finally, there's registration. Again, this was touched on briefly, but we have histology slides that we generally don't have whole brain. We need to somehow manage to shoehorn this oftentimes into a whole brain data set. We have to deal with absence of contrast, tears and wrinkles, things like staining artifacts. And these are issues that the techniques that we use in MR, brilliant alignment tools that we have, simply aren't able to handle these effects. So many people around the world are working on this. At our center, Istan Husar is working with Mark Jenkinson to try and come up with pipelines and tools that are actually appropriate to all of these challenges. And then finally, you have to try and integrate this with microscopy. So we heard a lot about polarized light imaging today. So this is a technique that was pioneered by Marcus Axer in Jülich. And basically, as Anastasia mentioned, you essentially have a polarizer that's rotating, and that causes the light intensity to vary due to birefringence of myelin. And then you can extract, for example, the orientations of fibers. Okay, so what I want to do for most of the talk is to sort of lay out for you a few of the different ways in which people try to use these data sets where we have both MRI and histology in order to ask specific questions. And I want to sort of emphasize the very different types of questions that we can ask. So if we start with a sort of classic MRI histology experiments where we'd be looking at, I'm just going to say, relationships and gold standards. So here's a good one. This is polarized light imaging of a gyrus, and you can see color is depicting the orientations, and you get this really beautiful fanning structure. Now, in that same brain, where we had the 0.5 millimeter resolution, we acquired this PLI data and then performed tractography in this gyrus. And what you can see is a well-known effect, which is called the gyro bias, which is particularly in the 1 millimeter data. You can see here that we have this bias toward finding connections really at the crown of the gyrus. And at 2 millimeter, we barely even are able to sort out the connections at all. Whereas at this very high resolution, we're able to actually depict in a really beautiful manner this fanning architecture. So this not only gives us a target resolution, it also gives us something that we can use, for example, to try to put constraints on our diffusion tractography that would enable us to, for example, do better, more at 1 millimeter resolution, for example. So another way that we might classically use MRI and histopathology together is, for example, here we're looking at quantitative susceptibility mapping, QSM, in ALS and controls. And you see in the ALS where we have these yellow arrows in the motor cortex that you have this hyperintensity. And this has been described in vivo. And so one thing that we might do, for example, is to compare in those brains then where we have that QSM data, same brains, across a range of ALS and controls, demonstrate that there's a relationship between QSM and ferritin. Now, we know to expect this, and we can demonstrate that we have this kind of a correlation. But really, at the end of the day, it's a very limited set of conclusions that you can draw from this. We don't know that the QSM signal is being driven by ferritin for sure. We know that histopathology signals correlate with each other, and we don't really know whether QSM is the best marker that we might have for ferritin, even if that is what's driving these changes. And so one of the things that I think we've oftentimes kind of kidded ourselves about is this idea that we're going to establish one-to-one mappings. And one of the things that my group is very keen on is trying to move away from that with the idea that we have a broad range of MR measures. We know that every one of those measures is probably sensitive to more than one thing that we might be interested in histopathologically. So trying to establish one-to-one measures may actually be missing out on a trick, which is that we could just try to establish this complicated set of relationships and take advantage of the fact that we have many signals with differential contrast, and they're telling us something. Now, what are the challenges to that? Well, one thing is that we can only make the histology measurements ex vivo, and most of the data that we would want to be making statements about is in vivo. And so one of the further complications is that we know that tissue in postmortem MRI is not the same as tissue in vivo MRI. And so actually mapping out these relationships could be a useful thing to do, but as you can imagine, it's a massive task. And so we're starting to explore whether this is even remotely feasible, again, using ALS as a model disease. But the promise would be that you could take in vivo MRI and try to predict from a broad range of multimodal set of scans what histology would look like. Okay, so really what this is saying then is that in the classic mode of comparing microscopy to MR, what we're doing is sort of actually a very sort of one-way, one-directional flow of information. We're using microscopy as a way to try to, in a very crude way, describe what MR is telling us. So what else could we do? Well, one way that we can try to make firmer statements is through the use of models. And oftentimes we think in terms of validation of models, but we should also think in terms of development of models. So this is some work that Jeroen Molink and my group published 2 years ago, where we had polarized light imaging data, again in this brain where we have 0.5 millimeter resolution. And one of the things, the first things that he did was to look in the corpus callosum. And what you can see is that both of the techniques are demonstrating that the callosum doesn't necessarily have this very simple fiber architecture, but actually exhibits somewhat chaotic pattern of fibers right at the midline, almost with a sort of braiding of fibers. And so what Jeroen did was then to ask, well, can we use this kind of data in order to tried to directly validate a fiber dispersion model that had been previously developed in our lab. And so the idea is very simple. You take your microscopy data, and you have then fiber orientations at very high spatial resolution, and you can then use that within to create essentially a voxel, an MRI voxel-sized description of fiber orientations. And so what Jeroen was able to show then was that was actually remarkably good agreement between the forward prediction of fiber dispersion from diffusion data and the dispersion that we measured from histology. In this case, this is actually a myelin stain. And the important point here is we're not just showing that they're correlated. We're showing that we can actually predict dispersion accurately. So it's not enough that we have a high R value here. The blue line there represents the line of unity. And so you can see we actually come up with quite good agreement quantitatively. So what's this giving us? Well, by sending the data from MR through a model and predicting the microscopy signal, we were not guaranteed to find some sort of agreement, even if there's an indirect relationship between the two. So by achieving consensus, we're able to achieve a validation. So can we do something even further different from that? So the last bit that I want to talk about here is a technique that we've been thinking about where we actually do joint modeling. And in this case, what we're actually trying to do is to say, well, we know that we have some amount of joint information between MRI and histology. We know that also each of these modalities has unique information. So can we build a model that actually tries to incorporate that knowledge into our analysis? So the example that we chose to go with, again, is looking at fiber dispersion. So here, this is just showing that you can have two very different systems. One is just a pure diffusion tensor. One is a number of sticks that are completely restricted, and you can generate the same diffusion profile from them. So what that means is that there's an ambiguity here, that dispersion and radial diffusion in diffusion MRI are not separable. So what Amy Howard's been doing in our group is to say, well, if we have microscopy, and she's working with that same data set I was showing earlier with respect to dispersion, if we have microscopy, it is informative about dispersion, and therefore that allows us potentially to overcome this ambiguity. So what Amy's done then is to build a joint model that looks like this. On the sort of left-hand side, this is essentially a conventional spherical deconvolution, or CSD technique. On the right, what you're seeing is an additional set of terms that describe how our current estimate of a fiber distribution function would look in a 2D plane. And so the idea is that you're constantly building up new estimates, updating your estimates of both what essentially the diffusion profile of a single fiber looks like, as alongside what the distribution of fibers looks like. And by sort of continually optimizing and trying to find the ways in which you can match both types of data, then you're able to overcome this ambiguity, and that's shown here. So on the left, this is if we just use the diffusion data to try and estimate dispersion and radial diffusion. On the right, this is showing the joint model. And you can see that so this is MCMC samples, so all of these are sort of relatively good fits to the data, and you can see that the joint model is able to hone in on a much more specific pairing of radial diffusion and dispersion. So if you can then start to look into what these different fiber distributions might look like. On the left, the CSD technique is coming up with much broader, more dispersed distributions, where the joint model is indicating that actually those estimates of the amount of dispersion may be overestimates. And so this actually begins then to draw questions to whether we should expect to trust some of the methods that we use for trying to estimate what's called the fiber response function in CSD. And in addition, Amy's shown that it's not necessarily the case that we expect there to be a single fiber response function that's consistent across the entire brain. She was also able to show that if we look in regions like the centrum semiovale, where we know that we have multiple fiber populations, that she's actually able to extract some of these secondary fiber populations. So sadly, I don't have a slide on what she's doing now, which is really cool. She's getting a data set called the Big Mac. She's taken a macaque brain where we have in vivo fMRI and diffusion. We've scanned it for, again, about 7 days using a multi-shell diffusion technique, and so we have several shells where we have 1,000 directions, and then she's gone through and is getting whole brain PLI alongside Gallius staining. So what we're hoping to be able to do then is to use that kind of a data set in order to be able to lock down some of these relationships in greater detail, in order to be able, ideally, to use the joint model to leverage situations where we don't have accompanying microscopy. So what I want to do is ask my moderators whether I can get 2 more minutes to describe the, yes? Okay, all right. All right, I promise this will be kind of cool. So I showed this slide at the very beginning. And some of you may have realized that actually I've only kind of focused on the upper left-hand corner, on using microscopy to tell us more about MR. What I want to very briefly do is give one example of how we could actually maybe achieve this greater integration with things like population imaging and rodent imaging. So I've already told you briefly about the study that we did looking at the callosum. That actually led very nicely onto a hypothesis-driven study that we did now using a very different types of multi-scale imaging, which is going to the level of from individual brains to the level of populations. So you may remember me showing this at the beginning, where we have in the callosum this sort of complex pattern of fiber dispersion across the brain. When we went to, we hadn't actually seen this really reported before, so we had a look in the biobank data, and we not only see that pattern, but we actually see that it's highly variable across individuals. And so this kind of prompted us to think, is there a biological significance to this variation? And in particular, is there a significance that might relate to function in the brain? And so in the biobank, we have both fMRI and we have diffusion. And what we wanted to do is ask the question, can we take microstructural features from diffusion and use that to predict functional connectivity in the brain regions that are connected by a given fiber pathway? And so again, Molink did this. He did it in sort of 7,500 subjects from the UK Biobank. The beautiful thing about the biobank is it also is able, if you wait long enough, you suddenly get a replication data set pop up in the database. And so this is showing the percent of functional connectivity that we were able to explain just by this really very crude linear model where we're predicting functional connectivity from microstructure. And I'd be very happy to talk about the fact that these effect sizes are small, and I think we should be thinking about what this means. But what I mainly wanted to talk about is the fact that that sort of discovery led to a hypothesis, which actually then led us to say, well, actually, there's other sorts of discovery that we can do using this same data, which is to look at genetics. So we take that function and microstructure link, and we can then do a genome-wide association study on that. And one example of what we found was that that link between microstructure and function was related to what's called the DAM1 gene, which is part of the Wnt signaling pathway. And I'm sure there are some people in the room who are familiar with this gene. I previously was not. But it does relate to things like axonal growth and guidance. So that discovery then is leading us to the next set of experiments that we want to conduct, which is to go back into animals, where if we take a transgenic animal from, for example, that DAM1 gene, we can then try to see if we can find, again, using basically the same signals that we use in vivo in the biobank, to see, can we see that signature in an animal that has had that gene manipulated in some sense, and then go in and, again, do microscopy and try and understand what that's telling us. And I really think that this kind of virtuous cycle of discovery and hypothesis and using techniques that span many different scales and many different types of scale is something that I think is going to be really exciting to see happen as we have a lot of these big data sets available to us in the future. So, yes, this is where I think the future is, and I would just like to thank all of my collaborators, and particularly Saj Babdi, who's kind of my main conspirator in a lot of the studies I've been telling you about. Thank you.